Now, Bill, you really pioneered the whole concept of bioenergy sorghum. How did you kind of turn it into the so-called game changer that it's really become?
Well, a lot of it is just a little bit of serendipity and not random, but foresight in terms of identity. I had a knowledge when I inherited the program in 1995 of existing material in there that might work from that. Combine that with a student who had an interest in biomass and accumulation of carbohydrate in stalks and stems. And we worked on it for his Ph.D. and then subsequently got interested in the process. And then, long story short, opportunity met preparation. When prices went high and the nation became interested in bioenergy production from biomass, we were sitting on some germplasm that we had pre-developed. So that provided the basis for the program. And then from there on, additional funding allowed it to expand, and eventually led to the first commercial hybrids that were put out there in the teens, second decade.
So you're in the right place at the right time, so to speak.
Right place, right time with material prepared.
Yeah. Now, your program led to a whole bunch of different innovations. What are some of the uses that you see for them?
Let me ask you a question before we go. Are you talking about on bioenergy, or are you talking about?
I was going to talk to interspecific hybridization.
Oh, okay, gotcha.
Let me
all right.
Let me work that into the question.
Sure.
So your program really pioneered, innovated in the realm of interspecific hybridization. What's some of the uses for this innovation?
Well, originally we were interested in it to facilitate gene flow from wild sorghum relatives into cultivated sorghum in the hopes that we could enhance disease resistance or pest resistance or something like that. It turned out that that's not what we've actually used it for in some ways. One way we used it was in hybridization of sorghum and sugarcane and the ability to move traits between those two species. More recently, we've used it to actually move a herbicide tolerance trait.
Hold on, I'm going to ask that question again.
Sure. Go ahead.
Now, Bill, your program has really innovated in the realm of interspecific hybridization. In a nutshell, can you tell us exactly what that is and why this is significant?
So interspecific hybridization is a mechanism that allows us to make crosses beyond just the species level into related different species of the same genera or the next taxonomic classification of relatives. Why it's important is it allows us to use traditional approaches to move traits from one distantly related species to a more cultivated species where they might have desirable effect.
Now, you've mentored numerous graduate students over the years. What would you say is the most important quality that young plant scientists have to have in order to really be successful in this?
If I had to pick one, it would be the ability to work with people, the ability to function in a team, collaborate, deliver their component of the expertise along in union with the other collaborators that they're working on. Obviously, they need a basic scientific knowledge level, but the ability to adapt, to learn, and to interact with people is really, really important.
Now, your work's really had a global impact, and we're living in this interconnected world now where technology has us all tied together 24 hours a day, 7 days a week. How's that, it's a double-edged sword, but how has that been an advantage to you in your work to have that global network and to be able to essentially access people easier and quicker than ever?
Well, certainly. As a young faculty, you don't start out that way, but traveling internationally and working with people when I started and getting to meet people through more senior researchers that I worked with at the time really established a basis and understanding of the global nature of of the commodity, the industry, and how that works, and how people do things differently in different parts of the world with different genetics. Then you bring that full circle 25 years later in a program, and it's those experiences with people you've either trained or worked with over those years that builds you a network so that you can contact them and connect to them on issues or problems or challenges that you might have in your situation where they can help, or vice versa.
Before I let you go, we're here at NAPB in St. Louis. What was your award again? It was the
2023 Public Sector Impact Award.
So, Bill, we're here at NAPB in St. Louis. You are the recipient of the 2023 Public Sector Impact Award. What's the biggest impact you feel you've been able to make to public sector research?
Well, I think that in one word, educating students to go on and work in the industries probably writ large will have the largest impact from my time and tenure as a faculty member. You can point to bioenergy sorghum, you can point to specialty trait sorghums. Hopefully those have a legacy and continue to be used and developed. But I think the students that go out into all other commodities in many other countries, as well as the U.S. and Canada, and they'll be in this business a lot longer than I will be. And so that legacy is probably the most important for, I would say, dare say, any public sector faculty breeder.
Thanks for sitting down with me.
Certainly.